Hey everyone, welcome back to This Explainer. Today we're breaking down the incredibly complex phenotypes and genotypes of hyperkinetic movement disorders. We're literally turning dense clinical text into a highly visual masterclass. So if you've ever wanted to seamlessly connect an involuntary movement straight back to the exact molecular mutation driving it, you are absolutely in the right place. Let's get into it. Here's our quick agenda for today. 1. Hyperkinetic disorders. 2. Essential tremor pathophysiology. 3. Dystonia classification and genetics. 4. Huntington's disease pathogenesis. 5. Huntington's-like phenotypes. And finally 6. Paroxysmal dyskinesias. Let's jump right into section 1, hyperkinetic movement disorders. So when we label a movement as hyperkinetic, the absolute key distinction is that these are involuntary movements that are unaccompanied by weakness, right? Unlike hypokinetic conditions, think Parkinson's disease, where movements get stiff and slow, these disorders are defined by excessive motor activity. It's basically like the nervous system's internal brakes have completely failed. You end up with way too much movement rather than too little. Let's just make sure we've got our shared vocabulary locked in. A tremor is a rhythmic oscillation. Dystonia, that's sustained, patterned, twisting movements. Now contrast that with chorea, which gives you these rapid, semi-purposeful, almost graceful dance-like movements. If those choric movements become large amplitude and proximal, we call that fallism. Myoclonus brings sudden, brief, shock-like jerks, and tics are stereotyped movements that patients can actually temporarily suppress. Having this conceptual map in our heads is super vital before we start digging into the genes. Moving right along to section 2, essential tremor pathophysiology. It's absolutely crucial to clearly contrast essential tremor, or ET, against Parkinson's disease. ET is typically a high frequency, like 6 to 10 hertz, postural or action tremor. So you'll see the tremor pop up when the patient is trying to hold a posture or reach for a target, like trying to pick up a coffee cup. Interestingly, it's also famously relieved by a little bit of alcohol. Parkinson's, on the flip side, gives us a resting tremor that happens when the limb is totally relaxed, plus you've got bradykinesia, or that classic slowness of movement. Shifting from the phenotype to the genotype and pathology, we know about 50% of ET patients have a family history pointing to autosomal dominant inheritance. Now, we haven't nailed down a single exact gene yet, partly because it's so common in the general population, but the research heavily points to the cerebellum and inferior olives acting as a sort of faulty tremor pacemaker. Specifically, we're looking at potential degeneration of GABAergic Purkinje cells, complete with these things called axonal torpedoes. If you're wondering what that is, an axonal torpedo is this wild structural defect where the nerve fiber just dramatically swells up, which completely wrecks the electrical signal trying to pass through it. Let's tackle section 3, dystonia, classification, and genetics. Physiologically speaking, dystonia is a total derangement of action selection. You get these co-contracting, synchronous bursts of both agonist and antagonist muscles at the same time. Basically, the central nervous system loses its surround inhibition. Think of surround inhibition like a finely tuned spotlight of muscle control. In dystonia, that spotlight suddenly turns into a massive floodlight, recruiting whole muscle groups that aren't even needed for the movement. Clinically, patients get these painful, twisting contractions, but amazingly, they can sometimes temporarily relieve them with a sensory trick, or just antagonist, like simply touching the side of their face or the affected body part. When dystonia is the only thing going on, what we call isolated dystonia, we can actually map specific clinical phenotypes right down to monogenic mutations. Take a look at TOR1A and KMT2B. These are the heavy hitters for childhood-onset generalized dystonia. FAPE1, on the other hand, usually strikes a bit later in adolescence, very often presenting with vocal cord or hand cramps. We're also tracking newer genetic markers now, like VPS16 and EIF2AK2. Grouping them out like this perfectly highlights how molecular diagnostics are totally reshaping our clinical expectations. Now, to avoid diagnostic traps, we have to clinically separate those isolated cases from combined dystonias. These are situations where the twisting movements are co-occurring with entirely different neurological symptoms, like Parkinsonism or myoclonus. And that brings us to a massive clinical takeaway: the GCH1 gene, which causes dopa-responsive dystonia, also known as Sagawa syndrome. Spotting this childhood-onset variant is literally life-changing. These patients present with prominent Parkinsonism and diurnal fluctuations, meaning their symptoms fluctuate and often get worse as the day goes on. Tragically, because of how it looks, kids are frequently misdiagnosed as having cerebral palsy. But here is the kicker. This specific genetic variant responds dramatically and sustainably to low-dose levodopa. It's the perfect example of why mapping the phenotype to the genotype profoundly matters in practice. Let's dive into Section 4, Huntington's disease pathogenesis. At the absolute core molecular level, this fatal autosomal dominant condition is triggered by an unstable, toxic expansion of more than 40 CAG triplet repeats on the Huntington gene, which sits right on chromosome 4. The math here is brutal. The larger the number of repeats, the earlier in life the disease manifests. And this is driven by a phenomenon called somatic instability. The DNA repeats actually continue to expand over time within certain cells, which just accelerates the cellular damage and the clinical onset. So, how does that toxic genetic expansion translate into physical reality? Well, looking right here, it's devastating. You can clearly see the profound atrophy of the head of the caudate nucleus. Because the caudate forms the lateral margin of the lateral ventricles, when it gets physically destroyed and shrinks away, it leads directly to the massive ventricle enlargement you're seeing right in front of you. So, what is exactly destroying that tissue? The CAG expansion creates a toxic gain of function. The mutant RNA and those misfolded protein fragments literally disrupt cellular transcription, they wreck mitochondrial function, and ultimately they trigger widespread apoptosis, programmed cell death, of the medium spiny neurons specifically within the striatum. Next up, Section 5, Huntington's like phenotypes. Now, medicine is rarely simple, right? Sometimes you'll have patients presenting with chorea and cognitive decline, perfectly mimicking classic Huntington's, but they actually test negative for that CAG mutation on chromosome 4. When that happens, we pivot to Huntington's disease-like or HDL phenotypes. HDL2 is driven by mutations in the JPH3 gene and features ubiquinin inclusions, which are basically microscopic clumps of cellular trash the neuron just can't clear out. Then you have HDL4, the most common of this group, linked to the Tatabox binding protein or TBP gene. Interestingly, this is genetically identical to spinocerebellar ataxia 17, so these patients very often present with prominent ataxia right alongside their chorea. We also occasionally see C9orf72 mutations jumping into the mix, bridging the gap with frontotemporal dementia and ALS. Let's wrap things up with Section 6, paroxysmal dyskinesias. There are 3 distinct episodic types of paroxysmal dyskinesias that we need to cleanly separate. First, paroxysmal kinesogenic dyskinesia, or PKD. This one is triggered by sudden movement, like standing up out of a chair really quickly, and it's heavily linked to mutations in the PRRT2 gene. Second, we have PNKD, which is non-kinesogenic. These attacks just happen spontaneously. They aren't induced by movement at all. And third, PED. This one is triggered by sustained exertion, like running a long distance, and is often linked to SLC20A1 or GLUT1 gene mutations. Getting these specific triggers visualized side by side really helps lock in that differential diagnosis on the fly. By connecting the clinical phenotype directly back to the exact genotype across all of these hyperkinetic conditions, we are really staring right at the future of neurology. So the big question we have to ask is, how soon will our granular understanding of these motor circuits allow us to actually intervene genetically? How close are we to silencing a mutant allele before a debilitating tremor, dystonia, or chorea ever even begins? It's an absolutely fascinating frontier. I really hope this explainer has illuminated these pathways for you. Keep analyzing, keep learning, and thanks so much for joining this masterclass.